Hello, my name is Raiden Farmer, and I am a computer science major at the University of Louisiana at Lafayette. And today I'm going to be doing a presentation orally on CRISPR-Cas9. Have you ever wondered what it would be like if there was a world without genetic diseases? What if we had a fairly non-invasive way to cure cancer? What if people no longer had to suffer from diseases like sickle cell anemia? Well, such aspirations are the goal of an emerging process called CRISPR-Cas9, a powerful tool being examined by companies like CRISPR Therapeutics. But what exactly is CRISPR, and why is something so promising also controversial? The story of CRISPR began in the early 1990s, when scientists discovered strange DNA sequences in bacteria. These sequences, now known as CRISPR, were part of a bacterial immune system that helped bacteria remember and fight off viruses. In the years that followed, researchers realized that these sequences were linked to proteins like Cas9, which can cut DNA at specific points. In 2012, Jennifer Doudna and Emmanuel Charpentier made a breakthrough by repurposing this system into a gene editing tool. They showed that Cas9 could be programmed to target and cut DNA at specific locations, allowing scientists to edit genes with unparalleled accuracy. This revolutionized genetic research, leading to potential treatments for diseases such as sickle cell anemia, cystic fibrosis, and even muscular dystrophy. But with great power comes great responsibility. In 2012, a Chinese scientist used CRISPR to create the world's first genetically edited babies, which sparked global controversy. You might remember this, it was a big deal at the time. This raised serious ethical questions about the implication of gene editing, particularly in humans. Despite these concerns, CRISPR has already transformed agriculture, medicine, and biotechnology, and in 2012, Doudna and Charpentier received the Nobel Peace Prize in chemistry for their work. But how does CRISPR work? Think of it as molecular scissors. Scientists design a guide RNA, which is a small piece of genetic material, that targets a specific part of the DNA that they want to edit. This RNA guides an enzyme called Cas9 to the exact location where it cuts the DNA. After that, the cell tries to repair the cut, and scientists can either insert new DNA, delete harmful mutations, or make other changes. This ability to edit specific parts of the genome opens up possibilities for treating genetic diseases and even improving crops. Here I have a model to demonstrate this. This is a little printout of a normal DNA sequence and then a sickle cell anemia DNA sequence. You can see that this triplet right here, the RNA sequence is different than this one right here. So I have another little model right here. This is representative of the normal DNA sequence, and this is representative of the sickle cell anemia DNA sequence. So what CRISPR would do is it would guide Cas9 into this double helix, and we see that it would try to cut this out, and once it does that, when the cell is trying to repair its DNA, it would replace it, it would insert the correct gene, or the correct triplet, into that slot and end with a correct, healthy strain, which would essentially cure the disease. Looking ahead, CRISPR's applications are endless. It could revolutionize cancer treatment by modifying immune cells to target cancer cells more effectively. It could improve agriculture by creating crops that are more resistant to pests or better at surviving in extreme weather. CRISPR might even help fight viral infections like HIV, targeting viral DNA. It also holds promise for gene therapy, where genetic disorders could be corrected before birth or even in adults. But while the possibilities are exciting, public opinion on CRISPR is mixed, understandably so. Many are optimistic about its potential to cure diseases and improve lives, especially for those with genetic conditions. However, ethical concerns still persist, particularly regarding the editing of human embryos. The idea of designer babies raises fears of inequality and unintended consequences. Some are also concerned about safety, as the technology could be misused or have unintended effects on ecosystems. In conclusion, CRISPR is the most transformative scientific advancement of our time, or one of, at least. It could cure genetic diseases, it could improve agriculture, it could change the way we treat cancer. However, we must approach it carefully, balancing its immense potential with the responsibility, and use it ethically and safely. As we continue to explore its possibilities, we are on the cusp of reshaping the future. By handling this power wisely, CRISPR could solve some of the world's greatest challenges and improve the lives of millions. Thank you.